Tripura. Dr. Subhajit Devnath has obtained his PhD from Tripura University in the field of animal physiology in 2011. So, sir has very vast experience, I should say very vast research experience in the field of animal physiology. So, without taking much time, I would request Dr. Subhajit Devnath to start the session. Sir, the session is yours. Please continue.
Yeah. Okay. Okay,
Thank
fine.
you, sir.
Just hold on.
Is there any PPT you would like to share?
Yes, I'll go ahead with that. Just give me
Okay,
a
fine.
second.
Okay.
Okay. Uh, okay, so I shall again forward my thanks to ATAL AICTE for providing an excellent opportunity for an interdisciplinary kind of discussion where people with specific interest on various recent developments and technologies could share their thoughts as well as uh, recent developments in the field. So, as I have already said, could you please tell me whether the screen is visible?
Yes, sir, it's
Coordinator,
visible.
sir.
You're sharing mobile through mobile now?
Yes, but it won't make much a difference. But just tell me whether the dimensions and everything are okay.
Okay, it's okay, it's okay.
Is it nicely visible? Okay,
Yeah.
fine. So what exactly happens is, so the topic, and I also congratulate the organizers for organizing this particular topic, where we'll be discussing about precision agriculture and nanotechnology. So it is again a very much timely concept that the organizers have floated. So precision agriculture, what is the? I am very much sure that all these days you have several brainstorming sessions on this different aspects of it. So precision agriculture, when we are talking about, we are essentially talking about optimization of resources. Acha, am I audible clearly? Uh,
Yes, sir,
Organizer,
you're
sir?
audible, but in between some voice disturbance is there.
Okay, okay.
Sir, it would be
So,
better if the PPT could have been run through PC because we are finding another screen on the top of this PPT.
so now is it okay?
Yeah, it's better now.
So what exactly happens is when we are talking about precision agriculture, basically we are exploring opportunities to optimize our resources that are needed for executing the agricultural aspects to it. To be precise, we are talking about utilization of fungicides, herbicides, as well as nutrients in a precise way, so that there is no misuse as well as there is no overdose kind of thing. So many a times, what exactly happens is, let us talk about the applications of urea. So urea is mainly provided in the plants with several nitrogenous these things, so that the amino acids and rest of the nucleotides and everything can be formed. So that was the main this thing of nitrogenous nutrients, for example, urea. But many a times we are also very much aware that urea is basically a chaotropic agent, which has potential for destabilizing tertiary structure of proteins. So what will happen if there is an overdose of this? So if there is an overdose of a particular nutrient, it will have much detrimental effect on the whole agricultural this thing ecosystem, isn't it? So the precision agriculture concept is mainly designed in a way so that there is an optimum use of the resources. Na kam na zyada. Now, in that aspect, one more development that has been recently this thing, that is applications of nanotechnology for this precision medicine. Now, this particular domain is very, very important these days because there has been multiple developments in nanotechnology from various aspects of engineering, including agriculture. So what exactly happens is whenever a particular material enters into a nanoscale, a large scale change in its effect as well as various other aspects takes place. So it gets a completely new dimension when we make a particular entity at a new nanoscale. And in my lab, where we are trying to put these nanoscale based peptide molecules for multiple different applications. So initially what has happened, we have isolated some bioactive peptides and within nanoscale range, but we saw that there is a much difficulty in stabilizing these peptides. So I'll be coming to those points. So one of our objective was to stabilize these nanopeptides in order to make it more stable, so that rest of the other opportunities that it will bring about along with it can be explored. So in this today's topic, let us discuss about that. And one more thing that our lab is interested in within our limited means is to establish a platform for simulation of nanoscale materials, so that the cost of the overall experiment becomes significantly reduced. So what exactly happens is, in order to design the whole experimental procedure as well as scaffolding the nanopeptides, it may happen that 10 different approaches should be there. So initially, for example, somebody is using ultrasonic vibrations, and somebody is using some kind of chemical inducers and so on. We do not know which will give us the optimum result. So in order to find out the optimum result, we try to simulate the whole procedure in a computational platform, so that it gives us much more flexibility to identify which method will be best for this particular peptide. So that was another objective of our lab. So let us go to the discussion. So as we all know that nanotechnology encompasses the understanding and control of matter at a dimension between 1 and 100 nanometers. Okay, at those dimensions, materials may have unusual physical, chemical, and biological properties, as well as functions that are remarkably different from those observed. So as I have said, example, that when you are spreading urea in your field, you are not only providing nitrogenous nutrient, you are also adding a chaotropic agent in the soil. So what would be its detrimental effect? Is there a possibility that we reduce the detrimental effect of urea? So that nanotechnology will help in a big way. So what exactly happens is, these are the peptides that had been identified by us and isolated in a pure form, which you can see at a scale of 10 to the power minus 7 meters to 10 to the power minus 9 meters. So well within the nanoscale range. So what was the activity of these peptides? Initially, these were antimicrobial peptides, but we are planning it to put it in multiple different usage. So that will be coming to this discussion as well. So once these peptides have entered their nanoscale range, their activity no longer remains only antimicrobial. Some additional activities are also seen. So we are interested to explore that thing within our limited means. So let us discuss about the formation of these nanoscale entities. So there are two strategies. One is bottom up and another is top down, as you all know. Now in bottom up approach, what exactly we do is, there is a chemical synthesis part, and the particle molecules will be arranged, and subsequently they will be formed within nanoscale range. So in this case, what has happened in our case, we followed a bottom-up approach, because these were naturally occurring low molecular weight peptides, which can also be synthesized. But however, at the first place, we have isolated them from a natural environment by controlled fermentation kind of, or controlled culture kind of technique. And then subsequently, we saw that these peptides have very low what we call molecular weight, because there are only 9 to 10 amino acids which are present in this, and eventually these low molecular weight peptides, we try to put them in a self-assemble mode or some induced assembly mode where the problem arises. We explored many opportunities where we found that assembly was not possible by some particular method, but later on successfully we assembled them, and we tried to explore their biomedical role, not only for inhibition of infection but some other roles as well. So we are constantly working on it. So basically in our lab, this nanopeptide assembling is we are using that bottom up approach. Subsequently, this particular field of nanotechnology for precision medicine was also explored in a big way. So we also correlated with that particular theme, and we found out that there are multiple types of applications of nanotechnology for precision agriculture. So as you can see here, we are essentially having concepts of nanosensors. Now in nanosensors, what exactly people are doing is, I'm very much sure participants have already had sufficient idea about all this. So what exactly happens is there are several entities. Basically, these are catalytic proteins which perform some kind of redox reactions. So what exactly happens is these redox reactions are controlled by entities like ion transporters and so on. So whenever there are some kind of changes in the electrical polarity of certain dimension, it will have a change in its catalytic property. Eventually a colorless compound will become colorful, or a colorful compound become colorless. So eventually you can find out that some kind of chemical reaction is going on. So these nanosensors are very important in multiple different applications, including agriculture. Then there are applications of nanoscale fungicides in order to control fungal infections of various kind in various food grains, and as well as these are important for post-harvest technology where people are acquiring the food grain and trying to maintain its novel properties, nutritional values, and so on. Then there is applications of nanopesticides. So the multiple pests which are causing a lot of damage to the food grains during on the field and off the field should be minimized by using these nanoscale entities which will be acting as pesticides in order to reduce the pests. And there are other applications as well. For example, insect pest management, as I have said. Then this is nutrients. Okay, nutrient supply is again a very important aspect to it. As I have said already, that although these are nutrients, they are having drawbacks of these chemical entities. Each and every nutrient that are being used for agricultural purpose, whether it could be nitrogenous fertilizers or other fertilizers like phosphorus and other things, even the micronutrients as well. So the optimum range should be understood in a nice way, so that how much exactly needed range should be provided. There is no overdose of that. And so in order to do so, that nanoscale entities will have very good this thing, because the bioavailability becomes very superior when the same molecule is entered into nanoscale range. So these are different aspects where nanotechnology, and there are some more which are being explored every day, where nano technology can be used for precision agriculture. Now we have found out one thing, that peptide based nanomaterials can be used in each and every point which has been mentioned here. For example, fungicide, and we have isolated an antimicrobial peptide within a nanoscale range, and we have initially seen that the antimicrobial potency is so good that it can inhibit a a range of different pathogens, plant as well as other pathogens. So we can reduce the amount of pesticides, fungicides, as well as infection control methods that are being used in the fields every day. We can reduce the burden of these xeno compounds or xenobiotic compounds, if we say. So these peptides are also biodegradable, because once they have reached a particular point, they lose their activity, and so what will happen is there will be no burden, and then some kind of resistant, usually, which is a big problem in biomedical research fraternity, where bugs develop antimicrobial resistance. So that resistance problem is also not there, because it is not a single molecule or single entity. It's a small low molecular weight peptide, and peptides do not have any probability to bring about antimicrobial resistance in pathogen. So there are multitudes of applications where these nanopeptides can be used. Now the problem here is how we will fabricate them into a scaffold. So are there any advantages to make them into scaffold? So if we make them into scaffold, it becomes very easy to apply. So it can be entrapped into a hydrogel, the nanopeptide, and can be used topically in order to reduce contamination in high-end agricultural setups, like where the liquid medium is used for agriculture, hydroponics, where the care should be given to each and every plant manually. So in those cases also, we can develop it into a hydrogel based topical application based thing, where these nanopeptides can be entrapped into a hydrogel and can be applied for a long term basis and within a very sustainable mean. So there are, it has been seen that the nanopeptide, if we develop in a proper way, we can use it in multiple different areas of precision agriculture. So that was the thing that developed our thought that why not give it a try. Initially, we tried for only a biomedical application, but later on we ventured into those disciplines as well. So how we got the motivation for making these nanopeptides into scaffold? So it has been demonstrated over the years that if we can scaffold these nanopeptides, they become superior in their behavior as well as nature, and their stability also enhances. So we have used here the reports that has been published by Cavendish Lab of Cambridge University. This was the lab who are a pioneer in peptide scaffolding and peptide-based nanomaterial design. So there are multiple aspects to it, where a single peptide can be scaffolded into a variety of matrix, which will be used for different purposes. So eventually, we can also think of putting the explants for making the callus culture, and for germplasm preservation, as well as some precision agricultural biotechnological applications, where these peptide based scaffolds can be used to grow the explant and for tissue culture research. So those possibilities are also there. So this is also the lab which during the 2012 proposed that there should be a nanomaterial based vaccine against the coronavirus. At that point of time, it was not COVID-19; it was only coronavirus 1, which originated during 2012. So that was the time this particular lab proposed that there should be a vaccine and a nano delivery based vaccine, which should be helpful for eradicating this particular disease. Later on, this thing have not been materialized. It would have been materialized that it's then itself. So it may happen that this pandemic would have been avoided. Anyway, this is the lab which has pioneered this concept of nanomaterial based scaffolding, and they are making it into larger macromolecular entity to give them a whole new dimension of everything. So we have used their concepts many times. So this is again where this nanopeptide scaffolds has been generated, and for animal cell culture as well as plant cell culture, both people are using those things. So, for example, for generating somoclonal variations and for generating some stress resistant plants and all, a single cell culture is being proposed. Now, what will happen if we isolate that single cell callus and break down into small soma clones and put it into the matrix? Into a completely different application. So these are all in the pipeline, and later on we'll have just just so this is again how this peptide scaffold is being done, again from the Cavendish lab. So linear assemblies of peptides and proteins serves as basic structural units for macroscopic materials in nature, such as in multiple different applications. Here the applications is given in collagen of various kinds of protective this thing. But the same peptide scaffold can be used for multiple different applications. So here they have demonstrated how this peptide scaffolding can be done. So as you can see, the beta pleated sheets are arranged in a particular dimension so that a completely new scaffold or a structure, a macrostructure can be originated from the low molecular weight peptides, and which have multiple different applications. So here again, the concept of what you call is peptide scaffolding comes from the natural phenomenon only, where it has been seen that smaller molecules can become larger entities and larger molecules can become smaller entities. Now, this is a fundamental this thing for nanotechnology, where either we are using bottom-up approach or we are using top-down approach. These approaches are nothing but has been sufficient motivation from the natural processes. Although it's an animal cell-based concept, but these concepts are applicable in agriculture as well. So this is again one more process through which self-assemblies of peptide can be possible in order to make it into a scaffold, and later on it can be used for multiple different applications. So in our case, what was the thing was, we isolated the peptide from a particular fermentation broth, and then we purified it eventually initially by SDS pH and later on by chromatographic techniques. So we found out that it has excellent anti-pathogenic activity against several aerobic and anaerobic what we call pathogens. So initially we were very excited that this antimicrobial peptide can be used in multiple different ways. But what was the problem in our case? We saw that these individual peptides, which are having very, very low molecular weight, were not very stable. The overall shelf life of the peptides were very, very low. So if we use them for some coating with inorganic salts, for example, urea, superphosphate, or things like that, in order to make these nutrients more available to the plant, we were seeing that there is a problem, there is a technical bottleneck, I would say, where the peptides were losing their stability, and they were also losing their capacity to make themselves or to remain antimicrobial in nature. So what was the idea? The idea was, we have isolated an antimicrobial peptide, and now we are fusing it with inorganic nutrients. So there will be two things done in the same go. One, the optimum nutrient delivery can be done to the plants, and we can also able to reduce the possibility of saprophytic infections in the plants. The idea was very, very good, but the bottleneck was there. We were losing the stability of these peptides. So these peptides were good, but they were not stable. Once we put them in the field, we were losing activity. So that was a bigger problem. So eventually we saw that there is a huge scope if we make it into scaffold. So if we make it into scaffold, what will be the benefit? The benefit will be, there will be a superficial increase in the molecular weight, and eventually there will be a steric hindrance. Due to steric hindrance, the internal peptide core area will remain intact and it will have prolonged activity. So the problem that was low shelf life can be overcome by making them into a peptide based scaffolds. Now scaffolds are nothing but multiple strands of peptide, as we have seen in the earlier this thing, no? The parallel or anti-parallel sheets of peptides. So what will happen is the enhancement of molecular weight will eventually lead into enhancement of stability. So what we can do is we can able to sustain the prolonged antimicrobial effect as well as the delivery of inorganic substrates to the plants for a prolonged period of time in a controlled fashion, isn't it? So in pharmacy there is a concept called sustained release tablets. Now what is that sustained release tablets? It will go on, all on a sudden it will not make a big menace, and with time it will continuously release the content, and eventually what will happen, there will be an optimum this thing delivery of the drug. Then also there will be no overdose, drug or nutrient or whatever you say. So sustained release kind of concept can be nicely what we call executed by nanotechnology, and I'm very much sure that you are already having that view already. So what they do is I'll share with you a video that has been produced by Monash University, which proposes that what exactly happens when sustained release can be achieved in a very good and controlled manner. So the example that has been
Sir, you're not audible. Hello? Sir, am I audible?
okay. So was it
Hello?
was it audible?
Yes,
Yes,
sir, it's
sir.
audible now.
Now you're audible.
Okay. So what exactly happened is, as I have said, that the idea was that we were initially trying to make the nanopeptide as it is, but later on we found that this peptide is very unstable. Okay, so we isolated that peptide from a culture broth and then purified it by chromatography, and eventually we found that it is having excellent antimicrobial property against many aerobic organisms, including like you can say pseudomonas and many saprophytic what we call, shall I say, soil borne organisms like azotobacter as well, some varieties of azotobacter as well. So eventually what has happened is we have identified an antimicrobial peptide which have multiple different applications to it. But was the problem was that these were highly unstable. Okay, in the field trials they were losing their viability. So how we thought to use those antimicrobial peptides? We thought that why not combine it with inorganic nutrients of the plants, so that there will be a sustained released based peptide. based concept, and eventually we'll reach a level where optimum nutrient will be utilized by the plant without any overdose. So the overdose is a big problem, as I have said, and given you an example of the urea overdose, where it has been seen that the detrimental effect on the soil microbiota is due to the chaotropic effect of the urea, which instantly denatures multiple protein strands from their proper tertiary structure destabilized, and eventually the organism itself dies, isn't it? So it provides nitrogenous nutrients to the plant, that is fine, but there is also a drawback to it. So the concept of precision agriculture is based on this particular thing only, where we are trying to optimize the resources, and at the same time we'll enhance the growth of the this thing agricultural output. So that was the main idea. So we saw that there is a huge opportunity in this, but in the field trial we found that these proteins were getting destabilized day by day, and eventually many a times instantly there were no effect at all. So what was the problem? We found that the problem is with the stability. So how we enhance the stability? By scaffolding it. So after scaffolding it, what will be the effect? The overall molecular weight will be increased, and then its thermodynamical stability will also enhance, and eventually they will also able to release the content in a sustained manner for a prolonged period of time. So we'll now have a video which will give you an idea how exactly that targeted delivery kind of thing works in a proper way. Now this is an example which has been developed by the University of that is Monash University of Australia, but I'm very much sure that these same concepts are applicable in multiple different aspects of nanoscale delivery mechanisms. So I would request our coordinator, sir, please play the first video from your side and share the screen, so that the participants can easily see it. So I had sent you one email, coordinator, sir. I think you have already
Yes, sir. Yes,
received
sir.
it.
I'm sharing it.
Okay. Yes. In a mute condition, in full screen.
Is this the first video?
Yes, sir. It is the first one.
Not this. No, not this one. Next one.
Let me just unshare it. You are asking about the nanoparticle-based drug delivery in the fight against cancer. This is the title of the video you want me to play.
It could be the character of the video, but the title of the video is not relevant. But the content which is going on is very much relevant. So please play the video in full screen without any sound. So what exactly we are trying to do here? We are trying to focus on how exactly nanoscale-based delivery mechanism put in place. So why this is very good for precision delivery of either an ingredient or a drug. So this video gives a tremendous idea. I'm not sure whether you have already had it,
but I cannot this thing to share this video with you. Okay, so is this in the full screen? So what exactly happens is these are some cells which are under fluorescent microscope. Now the fluorescent microscope has a very much good beauty in it. What is the beauty? It can differentiate on the basis of fluorescent tags which cells are working properly and which cell is not. So you can see there are two kinds of cells. One are pink in color, which are fluorescent tagged, and some are green in color, which are rhodamine tagged. So rhodamine tagged cells are the cells which are the target cells or virus infected cells. It could be a tobacco mosaic virus or things like that. So what exactly happens is usually before necrosis, I'm very much sure the necrotic pathway is such that whenever a virus infects a particular cell, it will keep on infecting the other cells as well. So the first infection starts for a single cell. So here what is happening is the nano drug has been delivered, and now these are the clathrin coated plates which will do an endocytosis based delivery, where the drug will be targeted to the nucleus. Okay. So what here has been done is the drug was being ingested by a endocytosis based mechanism, and the drug is now delivered to the nucleus. You can see it is overcoming the endoplasmic reticulum and lot more other things, and now it has been internalized by the nucleus. Can you see? So how precise the delivery mechanism is? It is going and reaching at a particular point where it is needed to be delivered, not anywhere else. Okay. Now what has happened is, after a certain time and a particular time of acclimatization, the delivery vehicle will burst and it will release the contents. As you can see, the red droplets. These are the content or the medicine which will enhance and which will change the cell to enter into a phase of not necrosis but apoptosis or programmed cell death. So what exactly happens is, why nano delivery mechanisms are good. Okay. Thank you, coordinator sir. So these nano delivery mechanisms are good because it will enhance not necrosis but apoptosis. In necrosis, what will happen? The cell will die all by itself, and therefore it will release all its content to everywhere. And eventually, the infection will spread from one cell to another. So what will happen? You will see the necrotic lesions on the plant. It could be anything. It could be your tissue culture derived plant. It could be your natural plant which you have breeded for many generations and preserved the germplasm. One fine you see that there is a necrotic nodule and eventually your plant is dying, isn't it? So why necrosis? Because necrosis is a natural process, but in apoptosis you can program it. How you can program it? By delivering of nanoparticle based drugs or therapies. So what will happen? It will identify a particular cell where the virus infection has taken place, and eventually it will reduce the virus infection or spread of it by making it into a apoptotic granule, and therefore the cell will die by itself and it will not release its content exterior. So no more virus replication can take place, and eventually a particular targeted cell will die, isn't it? So this was the idea of nanoparticle based delivery, which is relevant for all kinds of flora and fauna. So coming back to this thing, that what exactly we were interested in. So we were interested in the same thing, and since we had isolated the peptide and which was very good at the initial trial, so we found that the problem is only with the stability. So if we can enhance the stability, we can produce and sustain release kind of effect when we amalgamate our peptide along with other nutrients, plant-based nutrients, and eventually what will happen? There will be a optimum use of the nutrient. There will be no overdose, and eventually there will be sufficient plant growth. So the same thing has been demonstrated later on. Okay. Now, how do we produce these scaffolds? So as you can see here, that was the initial peptide. That is a 9 amino acid long peptide, which is very, very fragile. And eventually we folded it into two, and then it started a self-assembly. Okay. Now there are different strategies to initiate these folding into a proper scaffold. So some people will use ultraviolet ray, some people will use ultrasonic vibration, and multiple. And there are some chemical inducers also, which will induce these chemicals to aggregate and make into a scaffold, so that the overall molecular weight of each and every peptide is increased and it becomes more and more stable. But the problem with us was our lab was very, very small and we were not having sufficient funding. Okay, I'm sharing with you the exact scenario that we had like 6 years back. So what exactly happened? There were no much funding and we were not able to constantly isolate these proteins because we do not have that superior facility to isolate by HPLC. We used to take help from some other our collaborators. So we try to reduce that burden and try to understand that can we able to simulate the process? Can we able to check whether which kind of strategy is good for us, good for this particular peptide? So later on, we ventured into the simulation strategy of peptide assembly. So at one part in our wet lab, our the isolation mechanism and the fermentation mechanism that was put in place was okay, but later on we suffered the problem, and eventually we thought that we should go ahead with a simulation strategy to find out whether we can simulate the whole process and can eventually zero in into a particular process which we will follow in order to make our scaffold. So that was the general description how peptide scaffolding takes place. Now, once we do the scaffolding, it can enter into different kinds of tertiary structure. They may become star-like entities. They may become missile-like entities or liposome-like structures, and so on. So these are the possible arrangements which are possible after the peptides has been assembled into a nanoscale scaffold. So these are all within the nanoscale range only. We will show you that what exactly we did. So what was the idea? What was the this thing long-term goal? The long-term goal was this. So there will be the entity or active ingredient which will be coated on the peptide scaffold, if you can see. So what it will do is it will have a sustained release effect, as well as at the same time, because the peptides are already established as antimicrobial agents, so there will be also an antimicrobial effect also. So it would have been a blockbuster, but there were some problems. So in order to enhance antimicrobial and pathogen control, there is huge research that is going on, and which is costing government a lot of fund. So that was the first report of 2015-16, I guess. I think on 2017, where we reported about the antimicrobial peptide. And later on, we entered into a simulation strategy. So in order to develop a whole-scale prototype, what are the steps? So the steps are empathizing, defining, ideating, and prototyping, and subsequently testing. So what we did is we did a simulation for rest of the all four strategies. We started from defining, ideating, prototyping, and testing. So all these things we did it in a simulation platform, which has helped us to reduce our cost to a significant extent. And now we are in a position to report our next this thing, where we have found out which will be the best possible strategy for scaffolding these peptides. So that was our preliminary report, where we isolated it from a fermentation broth and purified it, and did the X-ray crystallography, and eventually made it into the protein structures. And we found out that if we can energetically stabilize them, we can able to have more stable peptides with enhanced shelf life. So this was our initial model. So in this, we are trying to find the dimension of the peptides. So this is the singular peptide molecule. These are 9 amino acid long, and we have identified the two axes, the largest two axes, which are within the nanoscale range. That is, one is as you can see, that is 21 point. 21 angstrom, and another axis is 11 angstrom. Now this is the whole 9 peptide amino acid long peptide that we isolated from the broth, and we used a certain technique to make this model, and eventually identified the dimension. Now these are the single peptide. Now these single peptide, due to low molecular weight, these are very, very fragile, as you can understand. Okay. So later on, we try to put it into a hands-on session by demonstrating some molecular visualization methods that we use. So we'll do a demonstration also. So next, this is the duplex peptide. Okay. Now in duplex peptide is what? Duplex peptide is one peptide upon another. So two peptides simultaneously we have kept, and we again show what was the dimension of it. So you can see there are two largest axes that we have identified. And what was this thing? The dimensions. These were 19 Armstrong long. Both were within 19 Armstrong long. So when we made it into a duplex peptide, stability little bit enhanced, but not completely. However, it is within the nanoscale range only. We were also worried whether if we make them into scaffold, their size increases. If the size increases very significantly and it goes beyond the nanoscale range, then we may not have the capability to explore the characteristics which it would have within nanoscale range. So we were also very much skeptical, and we are constantly visualizing the dimensions that it is enhancing. So from single peptide to duplex, from triplex. So till now we have the ability to make three. That means the triplex capability, three peptides side by side, and making into a small scaffold. So this is the duplex scaffold, which is again 19 Armstrong in dimension. So in the next, we did it for triplex. Just okay, we'll do now. This is the triplex. Now in this triplex, what happens? Three peptide chains were kept side by side. Okay, and what was the dimension? Two dimension we found out 22 and again 17. So two axes are 22 Armstrong and 17 Armstrong. So this is after the triplexing. That is three peptides side by side. So Cavendish lab have the ability to make n number of peptide sequentially and make into a large scaffold. Like they are calling it as an artificial skin. But we are not having that capability, and we are not working on that aspect also. So if we can do like approximately 100 fibrils all together, it will also serve our purpose, isn't it? So initially the duplex and triplex has also shown a very good stability, and I'll show you how the stability enhancement we did. So these are the peptide scaffolds of that antimicrobial peptide that we isolated from the fermentation and which have demonstrated very good what we call antimicrobial property. Okay, now these are the energy landscape. Please see, these are the energy landscape that we quantified by molecular dynamics simulation by GROMAC, where we found that once the singular peptide is there, the overall energy is minus 38 kilojoules per mole. So what exactly it says is, according to the laws of thermodynamics, once the overall delta G is reduced to a certain extent, the stability enhances. Why? Because the reduction in delta G will also reduce the what we call as degree of disorder or entropy, and eventually it will increase the stability of the peptide. So initially, the single peptide, which was very unstable, had a delta G content of minus 38 kilojoules per mole. Okay. So subsequently, when we made into a duplex,
gives user a much clear and sharp image. Also, it
hello.
signal which can be used for
So when we made
Yes, sir.
such a procedure, sir.
also camera.
Yes, sir. Yes, sir.
Just make them.
तो ये बेसिकली हम लोग वेरीफाई और
Please continue, sir.
So what has happened? Now this is the duplex. Please understand. Initially, the singular peptide had a delta G content of minus 30 kilojoules per mole, and when we made it into a duplex, it increased significantly up to 119 kilojoules per mole. Can you see? So later on, when we made it to a triplex, that also well within the range of nano scale, and it increased significantly up to 150 kilojoules per mole. So there was a 5-fold enhancement in thermodynamic stability. So one can argue that the thermodynamic stability that you have calculated from molecular dynamics, it may not be practical. But I am very much certain that at least the enhancement of stability is very much well demonstrated by this simulation. So it gives us a go ahead. Otherwise, we would have closed this chapter and we were not doing it anymore. Since these good results, we are very much excited and we are going ahead with our strategy to stabilize these peptides by scaffolding. So these are the domain flexibility that has been analyzed by Brownian dynamics and which again demonstrate, as you can see in the bottom. If you see in the bottom, the domain motions are very, very high. Okay, we call them R square motions, which are very, very high, the bottom one. Subsequently, when we made into a duplex, the overall dynamics of the peptide has reduced to a significant extent. And when we made it into a triplex, that is the top line, that is the top figure, as you can see, the domain motion were very, 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 very low. Only the C terminal end where had some exterior some extensive motion, and rest of the N terminal end as well as the rest of the protein was highly stable. So these are very good what do you call is what do you call some enhancing results which has helped us to go ahead with this scaffolding thing. And in order to make this bioactive peptide to be useful in multiple different domains. So now I'd like to request our coordinator again to please play the second video, which you initially played. So I just wanted to show how exactly the scaffolding takes place. So again, the example has taken from a published research, which has been published in ACS Nano recently. We have not reached that level, but this is how it happens. So would you please show the video second?
Is it the video one you want me to play?
This is not the video. This is already had it. The other video that please play that video. So the idea of this is to isolate a bioactive peptide which have a very low molecular weight and then make it into a scaffold. And scaffolding has different strategies how you will come with this. But in order to make this scaffold, you need to invest a lot in your wet lab. A 30 lakh rupees project you should write to a city and then get it done. So before doing that, why not? I did not given you this video, sir. Once again, you close it completely. All the YouTube videos that you have opened, you please close them and please go to the email. I did not think I have shared. You please go to the email, sir, and on the link. What?
Sir, could you please tell me which number video you want me to play?
Yes.
From your
The first video that you did in the beginning.
okay, I am clicking on the first link that you have shared with me.
Okay. So once again, please play, sir. So in YouTube, what is the problem is, if we do not click it, it will continue some other irrelevant videos. So I thought that you will download each of them, and then it would have been.
Sir, it is the video number 1 that you have shared with me.
We have already demonstrated it, but in the first place, when you played one more video, which I said to please close, that video, sir, I want. Not this. So please again go to the email. Okay, great. Now here, what is the scientist? What he has done is This report has been published in SES Nano. They make nanofiber which will self-assembling. Okay, so what you do is it is something like a chain reaction. Once you start the reaction, it will keep on scaffolding. So it may happen that the large scaffold you do not require, but it will go on. So you can see it. This is the initial peptide. The other lab, it is a different lab which have identified this particular peptide, which is having a different use, which is completely different from ours. But the strategy is similar. As you can see here, 2 peptide, 3 peptide, 4 peptide, like on, keep on, it is going. Can you see? So this is called the scaffolding. Okay, and this scaffolding within a nanoscale range will give tremendous results in multiple different aspects, in biomedicine, in nanosensor, and in precision agriculture as well. So that this particular lab from which we are showing you the video, they are not interested in precision agriculture, but we are. But the strategy is similar. This is how they are making a complete architecture of nanofibers and making it into a different kind of use, which is different from ours. But we are taking motivation from this, okay, so that we can bring about a different kind of solution altogether by putting the same strategy. So that was the idea. But okay, thank you.
So what has happened?
Okay, close.
So what has happened? Their strategy has worked for them because they have
Have a very good resource and they can do whatever thing they want.
But our problem is our resources are limited. So what we will do? Since our resources are limited, we will just simply sit. No, it was not that. So what we did is we did a simulation and trial so that we can find out
What will be the best strategy for us? Whether should we go with this particular peptide or not?
it will also happen while doing the
Scaffolding, it loses its activity completely and it's break down into pieces. This was also possible, but we saw that no, we can at least sustain up to 3 peptide long. If it may not be a very large peptide,
but it could be at least 3 peptide scaffold.
so coordinator sir, once again, I'll request you to share the next video, which demonstrate that how exactly we understood the overall stability problem. So again, the video would be played in a mute condition,
Where we have used a
simulator to see that how exactly the behavior of this stable and unstable proteins takes place. Okay, now this is the complete architecture of the triplet. Okay, so once we put 3 different peptides, what has happened is okay. So it could be a different result, but what exactly happens is we are using the same strategy. So this is the complete architecture of the peptide. Now we have to find out which peptide is more. So we have said that the tertiary peptide is highly stable than that of the secondary peptide. So there should be some proof of it. So what we did, and this is the complete because it was another demonstration. So as you can see, the complete energy landscape of the protein, isn't it? Some areas are white, some areas are red, some areas are blue, isn't it? So red areas are those which is highly negatively charged. That means negatively charged proteins are there, amino acids are there. Okay. Now this is the simulator in which we see the 2 different what do you call stability of 2 different peptides. Okay. So in both of the simulator, we have loaded our peptides. One is a dipeptide and another is a tripeptide. However, both of them will look like the same, isn't it? You cannot able to differentiate in a big way that how these are different. So now what we are doing is we are making it into a 3-dimensional model. Now this is a 2-dimensional model. We will make it into a 3-dimensional model. Okay. So one, and in the other protein also we will make it into a 3-dimensional model. Okay. So now what we will do is we will estimate the free energy on the whole protein. So for that there is also a this thing command line from which we can identify the free energy landscape. So what we will do is we'll change the pattern into the free energy landscape. So okay, now in this first protein, the energy landscape has given us an idea that it is completely different in color. Can you see? But in the right side, you see a completely blue color. Okay. Now, blue color indicates instability of a peptide, because what happens is this particular software, we call it Rasmol. It's a small software, 25 MB only in size. It can do tremendous. What it can do is it can color each and every atom on the basis of the free energy content, that is delta G. So it has been programmed in such a way, if delta G is increased, it will make into a blue color dot, and if the delta G is less than certain point, it will make into a other color, other than blue. So what exactly we are having is, we are having two peptides. One is highly stable, which is having multiple different colors, and another is highly unstable. So in a first phase, you cannot differentiate between the proteins. You will say that both are same in structure. Their overall structure may look same, but their stability is different. Why the stability is different? Because one peptide is tripeptide and another is dipeptide. So what exactly we are trying to show here is, on the basis of the scaffolding, the stability of the peptide is also enhancing. So once we enter into a phase where we are having a large scaffold within a nanoscale range, we can entrap our fungicide, we can entrap our herbicide, we can entrap our some antibiotic and so on, as well as nutrients like urea, superphosphate and so on, which will eventually deliver the content in a particular targeted way. Okay, that was the overall idea. So this is how we have found out that how stability can be enhanced, and we can also quantify how much stability has been generated by the scaffolding. So thank you, coordinator sir, for sharing it in a perfectly the way I wanted it. Sir, now please share the next video, so that we can again show you how the domain motion of the entities are there. So as I have said, that we studied one more thing, which is called Brownian dynamics. In Brownian dynamics, what we do is, we not only identify the delta G content, we also see the overall domain movement. That means what we do is, we shake the peptide. There is an algorithm called shake. So what we do is, we shake the peptide and we try to see what are the fragile entities, or what are the fragile part of the peptide. Okay. Now here you see, this is the first video of the singular peptide. Okay. Coordinator sir, I am audible, no?
Yes, sir, you are
So
audible.
what is here happening is, you can see only the some strands. Can you see? You are only seeing some rod-like things which are jumping in each and every coordinate. So what is this? So what we did is, we simulated the singular peptide in Brownian molecular dynamics, and we saw that most of the peptide is broken down. Only some strands are there. So pura jo protein hai, it has become denatured, because what exactly happens is, due to extensive motion of the proteins, when we put the thing in real time situation, what will happen? Because in the environment, there are multiple different forces that are acting on the protein, isn't it? There are hydrophobic forces, van der Waals forces, ionic concentration, pH, and so on and so forth. So what they'll do is, they'll break down the protein into pieces. So if your stability is not very good, this protein has no use. Okay. So what here happening is, the complete protein is broken down into pieces. Only some part you can visualize. Okay. But now we'll see the next video. Sir, please play the next one, where you will see that significant enhancement, although not completely stable, but significant enhancement has been done. So, sir, please play the next video. Okay. Now here you can see the beta sheets, the alpha helix as well, isn't it? So what is happening? The same type of protein in a duplex, now we are having much more stability. So from where the stability has come? So what we did is, we simulated the protein and we opened one more window where we have taken screenshots of every 15 picoseconds, and then we made it into a video. This is from the same software only. So it has given us an idea that where are the fragile fragments and where are the stable fragments. So in this video you can see, because it's a duplex protein or a duplex peptide, where two subsequent small peptides has been arranged as a scaffold. So here the stability is increasing. Previously, Sirf, you were seeing the rods and only the fragments of the whole protein. But here you cannot see the whole protein, but substantial part is still intact. So what does it mean? It means that the stability is increasing. So if we can achieve a complete stable peptide scaffold from this bioactive peptide, which we have already reported to have very good antimicrobial property against aerobic bacteria, so it will be a tremendous job that is done, isn't it? So what we'll do is we'll encapsulate the targeted nanomaterial, which may be prepared by our lab or by our collaborators, and then put it into the field trial. In the field trial, what will happen? There will be much more what we call stability, isn't it? So that was the general idea. So all over again, we'll go to the discussion phase where we have come to this thing. So okay, that was the idea, isn't it? So these were the things that we have demonstrated. Okay, these were one more peptide. So two different peptides were there, but similar to them. So eventually, we also found that. Similarly, we also found that it is also very much difficult to put everything in a single simulator. So in one simulator, we used to doing the visualization. In another simulator, we were doing the dimension estimation. In another simulator, we were doing the energy landscaping. So multiple different softwares we had to use. So we thought that why not put everything in a single space, and then written our own codes, and eventually kept our first thing in 2020. And we are also taking our collaborator particular innovation of about to also get the grant. It's the first patent from and the Indian. And these are we are to them for our monetizing it, and we are also doing customized training of this particular tool that which will do everything in one go. And these are the MSMEs. Some are government and some are NGOs to whom we are discussing regarding how to monetize our concept and of the simulator first, and then later on the other developments that we will do. And recently we have also demonstrated our this thing to IIT Mumbai, and the first phase is clear, and we are anticipating that we'll get into the second phase also of the innovation strategy summit, so that we can get some more funds. So that was the idea of whole perspective into one more idea. Labs. We are also looking forward to collaborators among participants as well as institutes. The major goal is to reduce the cost of various these things, so that uh, so
Sir, your voice is not clear.
so that was the idea. Of this outreach activity to collaborate with like-minded labs, and so by and there are some specific questions. If we can answer them, we and
Sir,
elaborate on some
you're
other
not
You still not audible, sir?
Sir, you're not audible actually.
Just
Am I audible to you, sir?
1 second.
Okay.
Coordinator, sir?
Yes, sir.
Yes, sir, you are audible.
You are audible now. So we could not listen to you at the very end of your presentation.
Okay. Okay. So that was the idea that the outreach activity let us to collaborate more so that eventually we can first play on not only this aspect, on multiple different aspects as well. And we are very much thankful for ATAL AICT
For giving this excellent platform where we can do extensive collaboration and outreach activities. So if there are some questions from the participants, we will try to answer them.
Okay, now I request participants to ask your queries if any. I must say, sir, that you have worked very hard and prepared so nice presentation. I can see that you have put our name on the header and footer also. So, can we get the copy of this presentation for the participants?
Yes, no problem. We can send it.
Okay. Thank you so much, sir. I don't see that participants are asking any queries or questions here. So, in that case, yeah. Do you want to share any quiz also?
Yes. Coordinator, sir, what was the architecture of the participants? All of them from agriculture or
Yeah,
some
okay.
other background
Yeah,
also?
mostly we are having the faculty participants. These are the faculties from AICT accredited institutes from all over the country. And luckily we got the participation from all different branches. Like we are having the electrical engineering, electrical engineer faculties, then mechanical
I
department faculties are
see.
there, civil department faculties are there. Even we are having the faculties from biotechnology, pharmacy, and some students are also there. PG
Okay.
students are also there. So we have a very diverse,
Great.
yeah, participation here. So I must
So I
congratulate and
thought
thank you.
we
Yeah.
understood some question from some people. There should be some question, I thought, and it becomes very nice if there are some discussion.
Oh,
So
okay.
if somebody
What we
want
can
to
do?
share
Yeah.
about,
What we can do? We
Hello,
can
sir.
ask the participants
Hello,
to kindly
hello,
write.
sir.
We have one query
Sir,
from the
yes,
Ajit Kumar sir.
sir.
Go ahead, sir.
please,
Myself,
please.
Ajit
Ah,
Kumar. I'm asking a question related to the uh, drug delivery. You explained related to targeted drug delivery. But when we use some
right.
nanomaterial, the material, how
Uh-huh.
the material can eliminate from human body because the metallic, some silver, gold, material which we
Great.
target the cells. But how
Yes,
we
yes,
can
yes.
eliminate them? Okay, thank you.
Yes, absolutely. Fine. Great. Let me be precise for this. Okay. So when we talk about a nanomaterial, most of the labs, because this is very easy to perform and easy to publish and do nicely. So what happens is whenever we talk about nanomaterial, we are essentially, but by and large, talking about nanometallic compounds. Okay, so I give you one example. So what we do, we take a solution of silver nitrate or silver chloride and then use some green chemistry into it. For example, some phytochemicals. So when we make a silver chloride solution, so silver remains as a oxidized particle with chloride. So what we do is we add some antioxidants in the form of plant derivatives, flavonoids, alkaloids, and so other things, vitamins, and several other things in a plant material, plant extract. So what we do is we show most labs you will see anybody who's working on nano, you will see that they'll demonstrate this kind of technology, and they'll generate nanoparticle by green synthesis. Okay. So the internet is full of this kind of studies. So there is nothing wrong with that. The problem is with the applications of it. As you have rightly pointed out, sir, I must congratulate you that you pointed out this. Would you please tell me which background you are having? Are you from biotechnology or some other background?
I'm biomedical engineer, as well as a PG in nanoscience and technology.
Okay, great. So most of the time this thing happens. Okay. Now these inorganic compounds have some problem. So let me give you one more perspective. Till today, any metallic compound that is used as a medicine is cisplatin. So you all know cisplatin as a compound for anti-cancer therapy, isn't it? Rather than that, there are very limited scope of using metal particle for drugs for therapy. Okay, other than some nutraceuticals. For example, some metal in the form of boron, then copper, and so other things are used as vitamins and mineral supplements for human consumption, but not as a medicine. Why? Because because it has been seen that there are multiple different problems to it. For example, silver itself. Silver is good, but there are some toxicity against it. So once in a nanophase, it can happen that it will do a targeted drug delivery. It will kill the cancerous cell, but eventually it can happen that silver will get accumulated in the body and then silver toxicity will arise. So the problem that you have pointed out. But we should also understand that there are 4 dimensions of nanotechnology. Why not we use peptides which are biodegradable? You do it, for example, like a targeted drug delivery through monoclonal antibodies. I hope that you are aware of it. In monoclonal antibody targeted drug delivery, what happens? The monoclonal antibody will reach a particular targeted tissue and deliver the medicine and kill the particular target. And there is also no contraindication because the monoclonal antibody is a protein itself and it will get digested in the body itself.
Okay.
Isn't it? So whenever we are talking about nanocompounds, why we are only focusing on nano silver, nano gold, nano this and nano that. So these nano things can be useful for
liposomal. Okay, sir.
Topical application. Liposomal based delivery is also very good.
Okay.
Yes, right. In liposome mediated delivery.
65 की strength है total science की. Now total. Fourth semester में 65 है total.
अब
हाँ. आज तो 24 जने आए थे.
I'm not getting what.
Okay, somebody has.
Anyway, so I
want to ask something. I think.
It's an excellent drug delivery mechanism where lipid coatings are being used, and in peptide also we can generate something which is similar to liposomes, but not exactly liposomes. Liposome is also very good because it is instantly resolved into the plasma membrane, and eventually the drug will be delivered. You are very correct in that respect. But why not we are talking about delivery of paracetamol in a nano range, isn't it? So why not you entrap paracetamol in liposome and put it into a nasal spray? So what will happen? Much less quantity of paracetamol will do wonders. Today you have to take 650 mg of paracetamol to get rid of your cold and cough, isn't it? Body ache and so on. Now you take 2 spray.
That's great, sir. Okay, that's great. Yes, that's great.
Are you getting my point? So
Yes, yes, yes.
called precision, isn't it? So we should also try to in India. You see, they are working on nano by putting nano.
Yeah,
This thing, what we
to
call
get
is
family man.
silver, gold, and some other things.
जाते जाते
Having very very limited scope. You can use for topical application. For example, you are having ring, isn't it? You are having a skin infection, fungal skin infection.
Yes, yes.
Uh, मतलब खुजली हो गया, ये हो गया, वो हो गया. In those cases, you can give this silver-based nanogels. Very good.
Okay.
But whenever we are talking about drug delivery, so think about the organics. So that was the idea. Just
Okay, is there any other query from the participants?
Okay, thank you so much for valuable points. Okay, thank you,
Thank you,
sir.
Ajit sir,
thank
for
you.
asking
Thanks.
this your query, and thank you, Devras sir, for replying so nicely. May I ask participant, is there any other query? Okay, in case there is
बाईस
no
हो गई
query, I would request Raman sir to kindly conclude
22
the session.
2022
Okay. Thank you, Dr. Suraji Devnath, for such a wonderful presentation and thought-provoking process. And also, I am thankful on behalf of whole our organization team to all the attendees here for attending the session very carefully. So once again, I am thankful to Dr. Suraji Devnath sir for taking out time from his busy schedule. Thank you so much, sir.
Thank you,
Now,
sir. Thank you.
Thank,
now we will
thank
be having dear participant. The next session will start at 12 p.m. and
12.
we will be then interacting with Dr. Saurabh Gupta. So I request all the participants to please join on time. Thank you.
Okay. Thank you, Raman sir. Thanks to all the participants. So you are again requested to kindly join in time. You can be here in this meeting, or you can leave and join again before 12 noon. Okay. I would request at least 5 to 10 minutes in advance. You all should join the meeting. Thank you so much. Uh, is there anyone asking something?
नहीं तो फर्जी वालों, कोनो जेसी बात नहीं।